Hello everyone, glad to have you here with us today. I am Jung-hoon at the Department of Biomedical Engineering at UNIST, and I am here to share with you an introduction on the fascinating possibilities that optical imaging provides for the understanding of life, especially in terms of the high resolution at submicron, or in other words, nanoscales. Let's start by considering how we can obtain information about the world around us in everyday life. We obtain information through what we call our senses. We can touch, see, taste, smell, and hear the world around us. Science and diagnosis of disease is also done through the same process. The only difference is that for more sensitive sensing, we have developed better technologies and tools to make the observation in faster and more effective ways. Now let's consider which of these senses gives us the most information. It actually depends on the target and needs, but in most cases, seeing can give us a huge amount of high-quality information to understand and diagnose the world around us. This is why you are looking at this video clip, to obtain useful information. Then what is seeing? Or in other words, what is imaging? It is literally all around us. Since the invention of the smartphone, everyone has an imaging system in their hands. You also have a built-in imaging system, your eyes. Common cameras are designed to see objects that our eyes can also see, and we call this photography. But there are other designs to make imaging systems that have different purposes. For example, telescopes are imaging systems that are designed to see astronomical objects that are too far for our eyes to see. Due to various reasons, the lens design is very different. As you can see here, instead of transparent transmissive lenses as in on a smartphone or a typical camera, telescopes typically use specially designed reflective mirrors to focus light. Microscopes have another different design with the purpose to see objects that are smaller than our eyes can see and typically look something like this. While these imaging systems look very different, they all operate on the same fundamental principles and allow us to understand the world at various different scales. Let's focus a bit more now on why optical imaging is so important to understand life and disease. I hope you noticed that something just changed. Yes, the color of this smartphone just changed, and you probably noticed it. This is because light has many degrees of freedom that can hold additional information. You can hold information in the color of light as you just saw, or in the phase, the polarization, or even angular momentum of light. In this respect, the color or the frequency or wavelength of light is very important for different sensing applications. To some of you, radio waves, X-rays, and light coming out from your monitor screen may appear as totally different concepts. However, it turns out that they are all something that we call electromagnetic waves. Light, or to be more specific, visible light, are electromagnetic waves with the wavelengths in the range between about 450 to 650 nanometers. Due to the law of diffraction, it turns out that the smallest object that you can image is limited by the wavelength that you use. So the smaller the wavelength, the smaller object that you can see. However, to observe life, we have to make sure that we do not harm or alter the sample that we are observing. For wavelengths shorter than the ultraviolet, the electromagnetic wave can kill the sample itself. For example, it can cause cancer. In comparison, visible light is very gentle on living cells, which gives us a nice window for a high-resolution observation of life. Optical imaging using visible light, covering the colors that we see in rainbows, and in many cases even beyond what our eyes can see into the near infrared, allows us to observe life with various contrast. Here you see E. coli bacteria that are expressing different color fluorescent proteins. You see a lot of information here in this image. You can tell that it is a beach with a tree in the background and the sun is near the horizon. This is how we encode information in creative art. We can do similar things to obtain information about life. Here, an artist did not create this beautiful distribution of colors. This is an epithelial cell that was stained to express different colors for different subcellular organelles. Mitochondria in red, nuclei in blue, and actin filaments in green. We can even go beyond single cells and make entire animals become fluorescent, customized to different needs. Combined with advances in biochemistry, new optical imaging technologies are continuing to be developed to probe life at various scales. With advanced technologies, we can observe life starting at the molecular, to subcellular organelles, to the single cell level, and even up to the tissue level. Unfortunately, due to the penetration depth limit of light, we cannot image entire human bodies currently using optical systems. You will hear more about other probes such as using ultrasound, X-rays, and MRI that are specifically aimed for this purpose in the following courses. We are here to see what biomedical optics can provide, so let's now see some videos of life in action. I briefly mentioned that the smallest object you can see is limited by the wavelength of the light that you use. This is a physical principle which, however, can be bypassed using some very ingenious tricks. Here is a kidney cell observed at such high resolutions, much smaller than the wavelength of light that is used, also called super resolution. Here, the endoplasmic reticulum, or ER, is shown in magenta, while microtubules are shown in green. This kind of high-resolution imaging of specific living subtargets is very hard to achieve using any other kind of method. If we now zoom in, we can see that the ER reshaping activities are highly dependent on the microtubule structure. This kind of dynamics can only be seen if we observe the moving dynamics at both fast speeds and high resolutions inside a living cell. Here, such power can be seen in a single subcellular organelle, the mitochondria, where the inner and outer membranes have been treated to express different colors. You can see that by increasing the resolution, previously unseen dynamic morphological changes now become visible. As you can see, optical imaging allows us to even image different molecular level contrast even within a single subcellular organelle, showing us the function of different compartments. The applications are not limited to single cells. We can observe collections of cells that make up an organ or even entire small animals. Here is a video of a Drosophila or a fruit fly embryo going through embryogenesis. While typical cameras do not have depth sectioning capabilities, we can make microscopes with 3D depth sectioning so that we can obtain high-resolution images of the 3D structure of biological specimens. Unfortunately, this projector and your monitor is 2D, so here we are showing two different perspectives to show kind of quasi-3D perspective. We can even optimize microscopes, in this case, to follow the 3D movement of the beating heart of a zebrafish. Here you can see the importance of speed and resolution in imaging, as we can follow the dynamic 3D movement of the entire organ while visualizing single cells. We have considered examples in high-resolution imaging from single cells to embryos or small organs in small transparent animal models. However, we are not limited to such small and transparent model systems, and we can apply optical high-resolution microscopy to even mammalian systems. For example, this image is a 3D rendering of neurons inside a living mouse brain. Although the mammalian brain is very untransparent or opaque, we can even control the shape of light that we use to obtain high-resolution imaging through such challenging environments. We can use such approaches to look at dynamics deep inside the living brain. For example, here are the response of microglia or immune cells in the brain that are reacting to damage. Here, let's take a quick break and see how these kind of experiments are actually performed in the lab. Okay, so here we're inside our lab where we're performing in vivo experiments on live animals. So here we have a special laser. So it's a near-infrared femtosecond pulse laser. So we're using light that's invisible to our eyes so that we can send it deeper into tissue while the sample is alive. Okay, so let's take a look at how the imaging setups really looks like. So in BME, we build real imaging systems. So you see that we use all these optical components. It's kind of like building a new Lego system. And so we're building imaging systems that are fit for our various purposes. So here we want to go as deep as possible in living animals. Okay. So let's go to the sample stage. And so here we have the mouse that's being mounted onto the sample stage. So we're gonna image this mouse's brain for this experiment. And so this mouse is alive. And so this mouse is basically asleep and anesthetized with a gas called isoflurane. And so here we have Dr. Seung-Hoon Lee with us. Okay. So after we set up the mouse, we put it underneath this objective lens, and we're going to be imaging the mouse's brain directly. Okay, so we have Dr. Seung-Hoon Lee performing his experiments. So we are imaging at the brain of a mouse right now, so the mouse is alive. So let's try to go in. So now we are going in deeper into the brain. So you now see the neuronal structure. And so let's go in deeper, and I think you can also, so we also changed the laser power to go in deeper. And so this mouse has some specific neurons that are expressing fluorescence. And for example, we can also use different mice that have fluorescence in, for example, immune cells or glia cells and so forth. And we can also image action potential of different mice using these kind of techniques. So let's try to go in deeper and see the soma structure. So here you see the soma of the nucleus, soma where the nucleus is located. And so we can all go in deep inside the mouse brain while the mouse is alive. Okay. Using light in biomedical optics does not stop at just observation. We can use light for various applications, such as treatment using light instead of the traditional way of using chemicals alone. We can also use optical forces to control and manipulate life at the nanoscales as well, which is nearly impossible using other types of mechanical probes. And movement of a micrometer-sized beads inside water. This highly precise and fast dynamic control of such small objects, and even simultaneous control of multiple objects, is only possible using optical tweezers. We can also use light to control the function of various cells. Here you see a video where a small worm called C. elegans is moving around. This particular worm has been engineered so that the muscular movements are inhibited upon exposure to specific wavelength of light that is flashing, as shown here. Using this kind of technology, known as optogenetics, you can use light to selectively activate or deactivate the function of specific cells, and even coordinate the firing of neurons, which are being actively used to help understand the brain. These kind of technologies have potentials to be developed into new treatments based on the precise and fast control of light. Imaging is a familiar concept that is all around us. You yourself generate massive amounts of images, which many big tech companies are willing to pay for to develop new functionalities based on the big data. In this respect, biomedical imaging is a representative field that is generating a great portion of the big data and benefiting from the recent advances in deep learning. For example, way on the left, here is a raw image that was captured on a microscope without any staining on the tissue. Now let's look at the two colorful images on the right. Can you tell the difference between the two images? It turns out that this one is the ground truth image that you obtain with the required staining. In comparison, the image in the center was generated using a deep neural network. The image far on the left was used as the input data, and this image was transformed into a virtually stained image. Imaging is the ideal source to generate such massive amounts of high-quality data because these networks often require those and is leading the current revolution in digital smart healthcare. So I hope you got a glimpse of what light can do for the understanding and treatment of life and disease. The field is very active and open with many questions. Let's keep imagining what kind of new cool stuff we can do with light.